Hello. So, in this class we will be talking about biomineralization. So, the formation of ferritin from the iron source from our food material or any other thing is also can be termed as biomineralization. So, within our body or some of the bacteria, so iron is stored in a definite form for the different functions, and those iron molecules are utilized afterwards for the synthesis of some important biomolecules. So, in case of iron, when we see that iron is present in the ferritin molecule, and the biomineralization process can be termed as the formation of biological iron oxides. So, we all know that how nature is giving us some mechanism such that iron ores and iron minerals are formed in nature and on the earth crust. So, in the same fashion, the mechanism of formation of biological iron oxides are pretty difficult, and there are basically some forms, and at least the most studied one we read here, there are 3 different forms. Because these forms are very important to know, because they are related to some of other interesting areas to know, that the biomaterials. So, how we get the nature is devising all these things, and if we can understand little bit of the formation of these materials and their typical functions, because some of them are magnetic materials, some of them are not, like magnetite and all these things. So, depending upon their simple crystallinity, how they are crystallizing into the system, what we have a typical matrix. The protein envelope, what is a typical natural one. So, biological part is there, your reaction vessel or the microreactor is nothing but your protein envelope, and within the protein envelope iron centers are entering, and that are critically crystallizing within that particular core. So, the different forms, they are basically controlling the formation of all these biominerals, how they are formed and their degree of crystallinity. Because their crystal structures are different. So, their crystal structure for this particular form, when they are forming one after another, that means they have different structures. The different structures. So, one such example for this material is magnetite. So, they are magnetic in nature, but they are formed in a typical environment which is little bit different, which is nothing but inside the lipid, the biological part which are taking the help of for the crystallization of these iron centers are lipid vesicles of some magnetotactic or magnetobacteria, because these particular things are also present in different birds which can fly from one place to another for navigational purpose. Which is therefore a navigational device for birds. So, they can feel within their body that magnetic part is there, and they can feel the corresponding direction when they travel from one side to another. Then another important material is ferrihydrite, which is forming from the protein coat of ferritin. Just we have seen. So, one is the lipid vesicle, another is the protein, and next is goethite. Which is alpha form of FeOOH, oxohydroxo iron, alpha form, and lepidocrosite, which is nothing but its gamma form. And these two are forming in another medium. So, matrix is different, the medium is different. So, they are formed in carbohydrate and protein matrix. So, it is a mixture of these two. So, this lepidocrosite is present in the teeth of some shellfishes. Shellfish. So, all this formation of these, that means whether they are lipid or protein or carbohydrate and protein mixture, not only the availability of the iron site, but some other thing, that means the other anionic form which is utilized for precipitating the iron in this particular form. If they are purely oxo and hydroxo, if no phosphate or other anionic form is not there, but the concentration or the gradient of these anion can control the corresponding structure and morphology of these species. So, the nature of the co-precipitating ions are important. Then we have seen that these lipid, protein and carbohydrate matrices are the corresponding organic substrates. Those organic substrates are also important, and those substrates are giving us typical boundaries basically, as we have seen the phosphate boundaries in case of ferritin formation. So, these are the inside coating and the boundaries. Then we can have some surface defects. So, if there is some defect, so defect will be there within the growth of this particular cluster molecules. Then the different inhibitors which inhibits the growth, that means which is taking away the iron centers which is not reaching there for the cluster formation or the agglomeration process. So, they are basically taking away or solubilizing those irons. So, they are functioning as typical inhibitors. And lastly, the two most important controlling factors, one is pH and the temperature. So, all these factors basically control the typical growth of this particular species, whether they will form the magnetite or ferrihydrite in a typical form. So, these are all corresponding iron biominerals, but little bit we should know, we all know from our childhood that what are there, but again we can recapitulate all these things, that how these materials are important. One is calcite, which we all know that this is nothing but our calcium carbonate, which is available in different cells in some octopus like species or octonia. And some, this octonia is basically a gravity device which basically controls our balance in mammalian ear, it is present. So, here basically the biomineralization process, which is not completely known like ferritin formation, because in case of ferritin formation most of the things are now known to us, that means how the protein is forming, how iron is entering, how the corresponding accumulation of iron is taking place, but not in the case of say calcium, not in the case of silicon. But we should compare the formation of this calcite or like apatite, which is giving us through this biomineralization process Ca2OH PO4 present in our bone and teeth. Then the fluoro version of this apatite, which is fluorapatite, calcium fluoride. And the growth of silica also in some cases in different cells is also important, and we do not know much about all these things, because how silica is accumulating into the system and how the corresponding fine structure of this silica material is also forming there. So, this particular part we have seen that how iron is getting stored, that means we can store iron or not only iron, but some important metal ions for their corresponding biomineralization process. So, the next part we will just go for the corresponding transport mechanism. So, the protein molecules again are important for the transport of iron. So, we have now the iron transport protein. We have stored and we have to transfer those iron centers from those ferritin molecule to the right site, where you can go for the synthesis of your hemoglobin or iron sulfur proteins or cytochrome molecules, and these are known as transferrins. And these are for human body or for higher animals only. In higher animals, because bacteria and all other small organisms, they follow different mechanism for iron transport. So, this particular molecule, the protein molecule, is strictly utilized for the transfer of iron, and these transferrin molecules are utilized for what? For the complexation reaction, the most important reaction we know. So, for complexation or for complex formation, which keeps the iron in soluble form, which is very important. So, once we have the iron in the soluble form, and then that particular iron we have stored in the oxo-hydroxo form, and which is in the insoluble form. Then again, if we take back those iron, we have to solubilize again, and some good groups or some good ligands should be available, which can bound to those iron and can transfer those iron from the ferritin site to the site of the synthesis of the important molecules. So, there are several such transferrin molecules are available when they are present in our blood. They are known as serum transferrin. When they are present in milk, they are known as lactotransferrin, serum transferrin, lactotransferrin, then ovotransferrin, and conalbumin, which is present in egg white. Present in egg white. So, all these molecules, these transferrin molecules, are basically monomeric in nature, and they are glycoproteins. Of molecular weight about 80 kilodalton. So, molecular weight can be determined also, and these are therefore proteins that contain what? That glyco means they have oligosaccharide chains. Which are also known as glycans. Greek oligos mean few. So, 2 to 10 saccharide chains are there, and which are ultimately linked covalently to the polypeptide side chain. Covalently linked to polypeptide side chains. And this particular mechanism is useful to control the free iron, which should be useful in biological fluids. Because these irons we have to deliver to the cells. So, some important groups should be available which can bind to this particular iron, and these transferrin molecules have very high affinity for iron binding. So, the formation constant for this complexation is pretty high, which is sometimes 10 to the power 23 mole inverse at say pH 7.4, which is also very important. When they are very strongly complexed, it is not possible to take out that iron by some other groups. Some other donor groups are also available in the biological system, but if your formation constant is not very high, it will only chelate it to that particular site, otherwise it will just go to the other molecules. So, when it is delivering those irons to the cells, it has two identical sites. For iron binding, two identical sites, and these two pockets had been identified, and that is why it can be considered as a dipheric unit. So, transferrin molecules are therefore the dipheric units. So, two particular domain is available where it can bind iron sites. So, again the most preferred coordination geometry will be followed here in the octahedral geometry of iron, and for knowing these complex molecules as a coordination chemist, what we always prefer to know, because we are always trying to locate the corresponding metal site and its immediate coordination environment. So, iron site is there, then the ligands, now our ligands will be the corresponding pendant groups what is available from the polypeptide chain, which is aspartate 60. 60 is your sequence number from the polypeptide chain, then tyrosine 92, then histidine 253, then again tyrosine 192. So, we have 4 positions fulfilled by the polypeptide chain. So, you have the sequence. So, the polypeptide numbering is from 60, 92, then 192, then 253. So, its coiling is there, and 2 other positions are occupied by a very interesting group, which is nothing but your carbonate anion. So, which can be considered as your co-precipitating agent, what we are just discussing. So, in presence of carbonate, and if you do not have the ligand system, what you get? You get the ferrous carbonate. So, when it is ferrous ion, it is only the ferrous carbonate, and ferrous carbonate can be precipitating out from the system. But in this particular case, the insertion of the iron within the transferrin system is due to the presence of that carbonate or bicarbonate anion. So, it is not only the carbonate, or sometime it can be a bicarbonate anion. Both of them, both the carbonate anion as well as bicarbonate anion can function as a very good OO bidentate ligand. So, these are from the carbonate or bicarbonate species. So, most of these, so this is phenol, tyrosine is phenol, aspartate is carboxy, and only one is nitrogen. So, again the iron center, and mostly we see, you can see that this particular thing is again in oxygen environment. So, only one is nitrogen. So, it is in oxygen rich environment. And the function of this particular carbonate is also important, because without this particular carbonate your iron cannot be strongly bound to that transferrin molecule. So, this has a typical synergistic effect. So, whenever the carbonate or bicarbonate anions are available, that means the basic iron carbonate or bicarbonate species as a whole is trapped inside the transferrin molecule. So, if you can have some mechanism where you can dissociate the carbonate or bicarbonate anion from the system, that means sometimes it is only controlled by pH. So, pH is the most important factor over here. So, if you can control the pH, it can be protonated in one step to bicarbonate and then to carbonic acid. So, when you take out the carbonate, your iron will also go away, because without this particular carbonate anion, that means when all the 4 coordination sites are there, but that particular arrangement is not very much stable, you must have this corresponding carbonate. Why? This particular carbonate is not only bound to the iron, and this particular fashion, which is not a very well known fashion of binding to a first transition metal ion like acetate. We know that acetate in bidentate fashion, it is not going to bind very nicely, because it is forming a 4-membered ring. So, 4-membered ring, most of the cases it can function only as a bridging motif. So, when they are bound to the iron center, and if we can determine, because the excess structure is known very well, we will find that one of the FeO bond is short and another is long. That means, it has some weak interaction with the second oxygen. So, basically it is functioning as a monodentate ligand. Since the other position is not available for binding by some other metal other donor atom, it is only occupying that particular position loosely. So, this carbonate anion has some other pocket, which is by the protein itself. So, the entire protein molecule has a binding pocket for carbonate. How? By some non-covalent interaction, because you have some free groups, free oxygen groups are there. So, this oxygen which is forming iron, and these will form some hydrogen bonding interaction with some other amino acid side chain, which is arginase. Which is ARG 121. So, we have arginase 120. So, this side basically, this side of the envelope of the transferrin molecule, we have another arginase residue. So, and that arginase residue number you can also find out is 121. So, that means 60, 92, then it goes to 121, then it is coming to tyrosine and then to histidine. So, histidine as the end number is 253. So, these interactions with this hydrogen bonding with the arginase residue of the polypeptide chain further stabilize the carbonate anion. So, this non-covalent interaction, only the hydrogen bonding interaction from the protein side chain can stabilize this particular small molecule within the cavity of this particular transferrin molecules, and this particular part is your guanidinium group. Guanidinium group. So, guanidium group of arginase is utilized for stabilizing your carbonate anion. So, we have a mostly oxygen rich environment and where you can have the corresponding carbonate anion, and that carbonate anion is stabilized by hydrogen bonding interactions. So, what we have in our hand is that you have to transfer the iron centers present in the ferritin molecule. So, this is your storehouse for iron in the ferritin, and this particular iron, when it is coming out as Fe2, and this Fe2 can be oxidized to Fe3 by ceruloplasmin Cp. The copper bearing protein molecule is available there. Ceruloplasmin, and once it is oxidized, this particular ferric ion goes to apotransferrin molecule. So, this apotransferrin molecule has two identical iron binding sites, and X-ray structure has proved that it has two typical iron binding sites. So, when it is taking this iron, it is transferring from apotransferrin to iron loaded transferrin molecule, and you have two such pockets what we just discussed for this iron site for transferrin molecule. So, you get that, and when this particular thing is forming, so accumulation of this particular iron site within the apotransferrin is further stabilized by a system where we have iron binding site, and one biological part is playing some important role there within a cavity type of arrangement, which is known as clathrin coated pit. So, these are basically clathrins. These are nothing but, you know the name, which are the receptor for transferrin. So, how we designate that? This is designated as TFR, receptor for transferrin. So, these receptors are playing some important role, because when this particular apotransferrin molecule is loaded with iron, they are sitting on this particular clathrin coated pit, because the clathrins are there. So, these some protein type of molecules are there. So, these are basically the clathrin, and these clathrins are responsible for closing of this pit and opening of the pit. So, when this particular iron is entering, so within this particular pit they are forming. So, at one hand you have the apotransferrin, you are taking up the iron, and that iron loaded system is there, and this particular thing when it is forming a circular form. So, ultimately when iron is getting entered, so you get the corresponding. So, you have the clathrin So, is basically it is giving a cell like structure, and within that you have the iron loaded transferrin molecules are sitting inside. Now, there is something that means we basically transfer this to the endosome. So, and the pH of the endosome we are just looking at how the carbonate binding is important for your iron binding as well as iron release, and how it is being checked by controlling the simple pH. So, the pH of the endosome is next reduced to say roughly 5 to 6 by a ATP dependent proton pump. So, because we all know that the ATP can control the pH of a particular medium. So, when ATP dependent proton pump is involved and ATP dependent proton pump is involved, and this particular proton which is available there is responsible for the protonation of the bound carbonate. Protonation of so, one transporter protein is now coming into the picture. Just you know the name, do not worry about this name. The name is also very simple, which is divalent cation transporter protein. There are several of them are present. So, DCT1 is coming into the picture. So, this DCT1 will transfer this iron out of the endosome into the cytoplasm. So, endosome was there. So, these are though not very complex biological term, but you should all know now that this particular DCT molecule then transports iron out once the carbonate is getting protonated out of the endosome into cytoplasm. Then it goes to mitochondria, and mitochondria transferring that iron to again back to the synthesis of heme type of molecules. So, how you utilize that particular iron now, or where we have started our discussion, or iron sulfur proteins for the synthesis of iron sulfur proteins. And if we find that immediately those iron centers are not required for the synthesis of heme molecules or these typical ferritin molecules, the excess of this iron can next be stored in ferritin. So, these particular mechanism which is solely dependent on iron coordination to the several donor atoms and the corresponding binding of the carbonate group and the corresponding control from the ATP, that means ATP proton pump can control the corresponding protonation of the carbonate group and it is getting released. So, pH is utilizing for the binding of the carbonate and sometimes this iron we are generating in our system. So, it is also in situ produced. The carbon dioxide what we are producing, those carbon dioxide during protonation giving you the bicarbonate anion as well as the carbonate anion. So, this particular ligand is playing that particular role, that it is utilized for iron trapping and binding within the transferrin molecule. Then if you just simply from one particular part to the other, if you can control the pH of the medium and that pH control can be utilized for the release of the iron in higher animals. But what is the case for this iron transfer for bacteria and small molecules, which you will find that in case of aerobic bacteria, we use some interesting molecules which are known as siderophores. Compared to your transferrin molecules which are pretty complex, these siderophore molecules are low molecular weight compounds of say about less than 1000 molecular weight, but they have very high affinity for iron. Sometimes their formation constant for complexation is varying from one source to the other to 10 to the power 30 to 10 to the power 50 mole inverse. So, these aerobic bacteria are present or they are originating from the different bacteria and fungi. So, this mechanism is useful for the microbes, that means the microbial cell to take up this iron, and so far a huge number of these which is greater than 500. So, 500 such species have been structurally characterized because they are low molecular weight species. So, molecular weight is less than 1000 because they are low molecular weight species. These particular siderophore molecules have been possible to determine their structures. So, they are structurally characterized at the same time. For their iron chelation. So, like your transferrin molecules, they also have the hard oxygen donors. So, once you identify that hard oxygen donor groups are available for iron binding and their iron chelation for the transfer in all these bacterial chelating agents which are siderophores, and one group of these molecules are their typical hydroxamates deriving from hydroxamic acids. They are hydroxamates, and hydroxamates are CONH-OH. Hydroxamic acid, from hydroxamic acid. So, you get, so when these two groups are binding to iron, so you get a OO donor bidentate part. So, all these molecules are very beautiful because they all providing very good organic ligands to bind your iron, and these hydroxamates when they are present, and those molecules which have been characterized are ferrichromes. And ferrioxamines. And another category contains catecholates. So, you have at the end point you have the catechols, example of that group is enterobactin. Also we can have carboxylates endpoints and phenolates. Together with other nitrogen bearing heterocycles or carboxylates. That means a combination of these two. In first case, when we have hydroxamates, all the three groups which are available for a octahedral iron center. That means, you need three such groups to bind iron site. In case of catecholates also, you have three such catecholate groups which are available for binding your iron site, and we get a corresponding octahedral iron site. That is why your corresponding formation constant is also very high. So, this particular molecules are isolated during 1952 or so, and one such very important molecule is your enterobactin. And which has in most of these cases either they are linear in structure or cyclic in structure. So, this is repeated on three sides. So, this is your whole structure. it has 3 parts. First part is your backbone, which is nothing but a triester. It is a cyclic triester, and which is very important to know there, because for binding of iron you have these catechol units are important, and these catechol units are important, and these catechol units forming a compound which is your FeL. The composition for the compound will be FeL. So, you have the triester part at the cover, and you have 3 pendant grooves basically. So, it is basically capping one. So, 3 pendant grooves are there. So, this pendant group, the second pendant group, and the third pendant group forming a octahedral iron complex. So, this backbone is important. Then the second part is your amide linkage, and third part is your metal binding unit. So, this particular form, how they are forming is a very interesting molecule, and it has given birth of so many synthetic studies on these type of molecules. So, this is one part, one O part, this is another O part, and this is the third O part. And if you consider the entire tricyclic, the cyclic triester part at the top, so which is basically connected, from this, and which is basically connected from this, from the top. So, you have 3 pendant groups are there. So, basically it is anchoring, is like a crane is anchoring. So, it is when they are, you have a very high formation constant, it is immediately taking up this particular iron from there, and when they are forming, so depending upon their corresponding orientation, it can form 2 forms, what we know, the delta form as well as the lambda form. When you view along the C3 axis of the octahedron, so both these 2 forms are forming there. But for some of the metal ions, because it is not only true for iron, but it is also true for vanadium, and for all some other system, and in marine organisms. So, this bacterial thing is also very important to know, because how we are competing with bacteria for iron. Bacteria also need iron for their regular survival, and when one form is forming, that means in some cases the delta form is forming, and in some other cases the lambda form is forming. And if we are able to hydrolyze this triester form, so that can immediately give us the corresponding iron, like the way we are getting the corresponding carbonates for the iron release from the transferrin molecules. So, because this triester backbone is not so stable for these siderophore molecules. So, all these things. So, next day we will see some other groups which are like ferrioxamines and some synthetic models, how good they are for iron chelation, and what type of these linear molecules, if we can have some medicinal importance, because if we can have some iron overload in our body, if we accumulate large amount of iron not only in our heart, spleen, liver or brain, whether we can take out those irons, and some of these groups, because they have very high binding affinity for iron, if they can be consumed as medicine, they bind iron and they can go out from our body. Okay. So, that we will see in our next class. Okay. Thank you.